So now let us see the comparison between these two cycles, that is menstrual cycle and estrous cycle. It can be written as OES or simple estrous is starting from Easter, E. So it can be estrous cycle also. Menstrual cycle takes place in primate females, whereas estrous cycle takes place in all non-primate mammals. In case of menstrual cycle, there is menstruation, and in case of estrous cycle, there is no menstruation. Menstruation means loss of endometrial lining in the form of blood flow, and whereas here nothing is lost. Why is this menstruation? Because endometrial lining is lost. And in case of estrous cycle, endometrial lining is reabsorbed. Otherwise, hormone levels and all those things are same during that particular period. The duration is different. This is a 28-day cycle or a 30-day cycle, and the estrous cycle duration varies in animals to a great extent. Now, those animals which show estrous cycle, they may show this cycle once in 1 year. Throughout this 1 year period, so this is say 1 year duration, in this 1 year they may have a reproductive season, which is the breeding season. So this is the reproductive season, and only during this season they get into this cycle, or they undergo these cycles. So during this period the females are to be or rather termed as in ester or in heat or in estrus, T-R-O-U-S, in estrus or in heat. Only during this time they copulate. That means the male will deposit the sperms in the body of the female and fertilization takes place. What about this remaining time, this time and this time? This time is non-reproductive phase, and it is known as, this is non-reproductive. It is called quiescent. Quiescent phase, quiescent word is used when the activity is completely lost or negligible. So here the reproductive system or its ability is totally reduced, and only in this reproductive season the female gets into this phase when she is termed as in estrus or in heat. This is the only time when they can copulate and reproduce. Rest of the time in the year they remain in quiescent stage, that is non-reproductive stage. Now some animals show one estrus cycle per year. So if there is one estrus cycle per year, then such animals are known as monoestrous. Cows, buffaloes, these animals come under this. So they are in this reproductive phase only once in a year, and that too in case of cows it is a very short period of time. So in cows, so example here is a cow, buffalo, all these animals. In cows it is only once in a year, and that too the duration is about 16 to 18 hours. So if the cow gets fertilized or her eggs get fertilized within this period of time, she would be able to reproduce. Otherwise, after this time the animal would again get into non-reproductive or quiescent stage. And again after 1 year, same phase will arise, that is estrus phase. Some animals, they reproduce twice in a year. So in that case, what would be the situation like? This is the monoestrus situation. In this case, suppose this is the entire year span. So animal becomes reproductive during one period and again during one season. So there are two estrus cycles, 1 and 2. Such animals are known as bi-estrus. So when animals have two estrus cycles per year, they are called bi-estrus. Sheep, goats, they come in this category. And third category is when the animals are in this estrus phase many times in 1 year. They are known as polyestrus. So many estrus cycles per year. Such animals are known as polyestrus. Examples of this: mice, rats, these smaller mammals, guinea pigs, they are all polyestrus. That means they reproduce 4, 5, 6 times per year, and every time they reproduce they get into this estrus cycle or they are in heat. This is the only time when they are capable of producing egg, and the chances are that the egg can be fertilized. So difference between menstrual and estrus cycle is only restricted to this menstruation phase, and in estrus cycle we can have or we can see animals with one cycle 1 year, 2 cycles per year and many cycles per year. So after this menstrual cycle, if we saw why menstrual cycle takes place, that if there is no fertilization. But if fertilization takes place, there is no menstrual cycle. So now let us take up the various changes or stages which take place during fertilization and after fertilization. So let us now talk about fertilization. So now let us understand what all happens during this process of fertilization. Though we write it as fertilization, which means fusion of gametes to form a diploid zygote, but it doesn't happen in one go, like just gametes come and they fuse. There are many things which happen. Before this fertilization actually takes place, when we wrote all the stages of sexual reproduction, the first was gametogenesis, we saw spermatogenesis and oogenesis. Then we took up these 2 cycles, that is menstrual cycle and estrus cycle, in case if fertilization does not take place. There is one more step before fertilization which we wrote, that was transfer of gametes. In case of human beings, it is known as insemination, where the sperms are deposited into the body of the female. The sperms are deposited into the vagina very close to the cervix. So now after this insemination, we will divide this entire process of fertilization into stages. The first stage is that the sperm is going to approach the egg. The sperm is deposited in the vagina. The egg is in the fallopian tube. Try to recall the structure of the female reproductive system. Sperms are in the vagina and the egg, after being released on the 14th day, is in the fallopian tube. It is going to remain in the fallopian tube for 2 days. So now the sperm has to travel through the cervix, uterus, the last part of fallopian tube into the ampulla region where it can fertilize the egg. So first step is approach of sperm towards the egg. How does it approach? Number one, how many sperms are deposited? When the sperms are deposited, it's the complete secretion which is known as semen. About 3 to 4 milliliters of semen is deposited in the female reproductive tract. It contains about 200 to 300 million sperms. These sperms are going to swim, but when they are deposited, it is in the form of a fluid. Semen is in the form of a fluid, and if this fluid flows everywhere, then the sperms are not able to swim properly because they need a medium plus some more additional support. So now what happens here is, if you are able to recall this composition of the secretion of seminal vesicles. In case of male reproductive system, when the sperms are going up the vas deferens swells into an ampulla, and just at that ampulla there is a gland which secretes its secretion. When we wrote about its secretion, we wrote couple of things like prostaglandins are present in that, plus some coagulating enzyme. So the coagulating enzyme of the seminal vesicle secretion. So we can write in the bracket from seminal vesicle. It coagulates the complete semen. So instead of remaining in the form of a watery, fluidy thing, it becomes little coagulated. Coagulation is like if you break the unboiled egg, then all that white part which is the albumin, it starts to spread everywhere. But if you boil the egg, this liquidy albumin, it starts to become little solid, it starts to solidify. It's not going to become completely solid, but it starts to become solidified. Same thing what we see in case of milk to curd formation. Milk is more fluidy, and then when it starts to coagulate, we get curd. Same kind of change is seen here. So the liquidy semen starts to coagulate under the effect of coagulating enzyme which is present in which is secreted by seminal vesicle. Why is this required? It is required because so that all the sperms, they remain together. If it gets little solid, semi-solid type, then all the sperms are going to remain together and their movement will be easier. If everything is pushed, it will be easier for them to move. So this is first. After some time, the anticoagulating enzyme of the prostate gland will again make it liquid. When it enters the uterus, so in uterus, the anticoagulating enzyme of prostate liquefies it. It is deposited in the form of a liquid. With the help of a coagulating enzyme, it becomes little semi-solid and then it moves from vagina through cervix into the uterus. In the uterus, again it is liquefied. So the enzymes which are helping are coagulating enzymes and anticoagulating enzymes. Now the sperms start to swim. The sperms swim at a rate of 2 to 3 millimeters per minute. In some books it is written 1 to 3 millimeters, in some it is 2 to 4. So, it is a slight range. So, approximately 2 to 3 millimeters per minute. It is the rate with which the sperms swim. The medium is liquid now, so they are able to swim. Do these sperms encounter any kind of barrier? They are going through the uterus. How is the lining of the uterus right now? Suppose it is all happening on around that 14th day when the egg is in the fallopian tube. At this time, the endometrial lining is thick under the influence of estrogen, and progesterone has just started coming in because corpus luteum is in the process of its formation. So, endometrial lining is thick. So, sperms encounter barriers. There are two types of barriers. One is physical, that is the thick endometrial lining, so they have to swim through those folds which are in between this endometrial lining. And second, there are chemical barriers also. Chemical barriers, the secretion of the uterus or vagina is acidic. So, secretion of, we can say, the female reproductive tract is acidic, and semen contains bicarbonate. So, these bicarbonates will also neutralize the acidic pH of the female reproductive tract. So, they have to encounter two types of barriers. Is there anything which helps these sperms to move? So, there is some helping thing also. Sperms are helped by the contractions of the female reproductive tract caused due to prostaglandins, by the contractions of vagina and uterus due to prostaglandins. These prostaglandins are already present in the semen. They are the secretion of seminal vesicle, and they cause the vagina and uterus to contract from lower side towards upside. So, it is sort of a peristaltic movement which takes place and pushes the sperms upwards. In this female reproductive tract, the sperms undergo one more change. It is called capacitation. Sperms undergo a change which is known as capacitation. During this capacitation, the membrane of acrosome, it becomes more permeable to calcium, because now what is going to happen is that the sperms are moving through the uterus. When they come to the fallopian tube, they have to fertilize the egg. Before they fertilize, they will have to digest all the walls, the corona radiata, zona pellucidum, all those layers to reach up to the egg. So, how will they digest it? With the help of enzymes which are secreted by acrosome. But for that, the acrosome has to rupture, and for acrosome to rupture, the membrane around it should become less stable. So, this capacitation is a change in the female reproductive system, changes in the sperm which takes place in the female reproductive system. So, let us write this is very, very important. Changes in sperms which take place in female reproductive tract. Because of these changes, the sperm attains functional maturity. What is the function of the sperm? It has to fertilize the egg. But for fertilizing the egg, it has to become capable of doing that. So what happens in capacitation is they attain functional maturity. Normally, it takes place in about 6 hours period. So this is like duration of this capacitation is 6 hours. So now the sperms are swimming. There are some barriers. There is something which is helping them, and they are undergoing some changes in themselves so that they are now functionally fit, capable of fertilization. So this is still approach. It is still approaching towards the egg. It has not reached up to the egg. So what all changes are taking place once the sperms are deposited? First, this semen is going to coagulate. It will become little semi-solid so that entire thing can move together. Helped by coagulating enzyme of seminal vesicle. Then once it enters the uterus, it again liquefies with the help of an anticoagulating enzyme secreted by this prostate gland. The sperms start to swim, and energy is provided by the mitochondria, nebuncon, which is present in the middle piece. So they swim at a rate of about 2 to 3 millimeters per minute. When they are passing through or when they are moving through the female reproductive tract, they encounter some barriers. There are some physical barriers like the thick endometrial lining, chemical barriers like the acidic secretions of the uterus and vagina. Then there is something which is going to help their movement. They are swimming on their own, but prostaglandins, which are also present in the semen, they bring about contractions of the female reproductive tract in the form of peristalsis. So, but it is starting from vagina going upwards in the uterus. So, this also helps the sperms to move up. When they are passing through this female reproductive tract, they undergo some changes. These changes are mainly in the acrosome membrane. The acrosome membrane is very stable and it becomes less stable so that it can now rupture. It has to release those enzymes which will digest the walls around the egg. So that is known as capacitation. The important thing is it is a change, or these are the changes which take place in the sperm when they are in the female reproductive tract, and the normal duration of all these changes is approximately 6 hours. Now the sperms are going to reach the fallopian tube. How many sperms were deposited here? 200 to 300 million. After all these things, how many reach up to the fallopian tube? This number is about 100. So only 100 sperms reach up to the fallopian tube. What about the remaining? They either get lost in this passage due to physical barriers they cannot cross, or because of chemical barriers they die, but they are not able to reach there. These 100 which reach, they are the best. The reason, they have overcome all physical, all chemical barriers, and they have also undergone whatever changes which were required in them. So the 100 best sperms, they have reached the fallopian tube. In higher animals, there is something called identification. The egg and the sperms, they have to identify each other. It is a species specific reaction. It is known as fertilizing, anti-fertilizing reaction. It is basically identification. The egg and the sperm, they have to identify. Fertilizing is found on the wall of the egg, on the egg wall. these are carbohydrate or mucopolysaccharide molecules. And anti-fertilizing is found on the sperm wall or sperm membrane, and it is made up of proteins. So just if we have to understand what exactly is meant by this identification, suppose this is the wall of the egg and the egg has these fertilizing molecules, say like this, just to understand what exactly it happens. It is very much similar to lock and key arrangement what we see in case of enzymes. And this is the sperm wall, and the sperm wall has anti-fertilizing like this. So they can fit into each other. Now here it is not exactly fitting thing, it is identification, and this fitting, when we talk of it, is actually the bonding. So if they are able to fit, that means they identify each other, they make bonds with each other, and then fertilization will take place. So this is a species specific process. This whole thing is a species specific process. And fertilizing and anti-fertilizing in a particular species are fixed, and that is why the eggs and sperms of two different species, they don't identify each other and they don't fuse. There are some exceptions where this happens, but they don't fuse. So this is identification. This egg and sperm, they have identified each other. We are still on the first process, that is the sperm is still approaching the egg. When it has to approach the egg, it has to digest all those layers around it. So now the sperm secretes, its acrosome is going to, and entry of sperm will always be from the animal pole where that nucleus is closer. So sperm acrosome secretes enzymes. These enzymes are known as sperm lysins. There are 3 sperm lysin enzymes. One, hyaluronidase, we will see what role they perform. Hyaluronidase, then acrosin, and third, corona penetrating enzyme. Three enzymes are secreted: hyaluronidase, acrosin, and corona penetrating enzyme. These enzymes are secreted from the acrosome, which is formed from the Golgi body, and the acrosome ruptures. Why is it able to rupture? Because it has undergone capacitation, its membrane becomes less stable, ruptures, releases these enzymes. Together they are known as sperm lysins: hyaluronidase, acrosin, and corona penetrating enzyme. Now let us see what these 3 enzymes do and how does the sperm actually enter. It is still approaching, it has not touched the vitelline membrane, and when it touches the vitelline membrane, that is when the next stage will take place. So we are still talking about how the sperm is approaching the egg, what all changes take place. Now it has secreted the enzymes, and now these enzymes will digest the walls. So how these enzymes digest the wall and how the sperm actually enters now. So that is the next stage. We are still discussing the first step, that is approach. And now in this first step, the sperm lysins, that is hyaluronidase, then acrosin, and corona penetrating enzyme. how do these enzymes work? Hyaluronidase enzyme digests hyaluronic acid, which is a mucilaginous substance, a gluey substance, which holds the cells of corona radiata. If you remember the structure of the egg, there is a vitelline membrane here and then there is zona pellucida, which is a non-cellular layer, and then there are cells, follicular cells, these follicular cells are held together by a mucilaginous substance which is actually binding them together. This is hyaluronic acid. So, the enzyme hyaluronidase, it digests or breaks this hyaluronic acid. As this binding substance is dissolved or digested, these cells, they get loosened up. And when they get loosened up, the sperm can move through them or in between them. So, this is the role done or performed by hyaluronidase. Acrosin digests zona pellucida. Acrosin digests zona pellucida. Hyaluronidase digests hyaluronic acid, which actually is binding these cells of corona radiata. Acrosin digests zona pellucida, and corona penetrating enzyme digests the cells of corona radiata. So, these two enzymes, that is hyaluronidase and corona penetrating enzyme, what are they doing? One, hyaluronidase is digesting the gluey material, and this corona penetrating is also digesting these cells. So, one, the cells are loosened up, then the second enzyme digests even the cells. Acrosin will digest this zona pellucida, and now the sperm can come in contact with the egg membrane. So, it is these three enzymes which are helping in approach of this sperm up to the egg. So, in first stage, that is approach of sperm towards the egg, we have to start from how many sperms are deposited, that is insemination, all the barriers, physical, chemical, capacitation, and then release of these sperm lysins so that the walls around it can be digested and the sperm can come in contact with the egg. Now, the next stage is when the sperm comes in contact with the egg. There are multiple things which are going to take place. So, now the next stage is going to be fertilization, and again we will have sub parts of that fertilization process also.